Hi and hello everyone, this is Ravindra Babu. Welcome back to my channel Silver 90s BIONEET. By the last video, we successfully completed the detailed mechanism of prokaryotic translation. Still, we need to discuss the mechanism of translation in eukaryotes. Here, the discussion of detailed mechanism of translation in eukaryotes is beyond the knowledge of this intermediate standard. So that here we are going to discuss the differences between a prokaryotic translation and a eukaryotic translation, followed by the concept of polyribosomes and post-translational modifications. Before we going to start this today's topics of central dogma of molecular biology, here a small request from my side: kindly watch all my videos made on this molecular basis of inheritance for the better understanding of the concepts, and like and share the videos and subscribe my channel. Hope you do this. Let's get into our last topic of central dogma of molecular biology, that is differences between a prokaryotic translation and a eukaryotic translation. So this is the prokaryotic translation and eukaryotic translation. There are few differences between prokaryotic and eukaryotic translations. One is the initiating amino acid. The initiating amino acid in the case of prokaryotic translation, you know very well, that is formyl methionine. What is that? Formyl methionine. But in the case of eukaryotic translation, the initiating amino acid is just a methionine. This is a basic difference between prokaryotic and eukaryotic translations. The another difference is the initiating factors. The number of initiating factors in the case of prokaryotic translation are 3. Those are initiating factor 1, 2 and 3. But in the case of eukaryotic translation, the initiating factors are more than 3. The discussion of eukaryotic initiation factors is beyond the knowledge of this intermediate standard. So just to remember, there are more than 3 initiating factors of translation in eukaryotes. And the third difference of prokaryotic and eukaryotic translations is elongation factors. The type of elongation factors in prokaryotic translation are elongation factor Tu, elongation factor Ts and elongation factor G. We have already discussed the functions of these elongation factors in the last video of translation mechanism. If you want to know this function of these elongation factors, you can go through that video. I will provide the link in the description. In the case of eukaryotic translation, the elongation factors are different, but the elongation factors perform the same functions done by the elongation factors in prokaryotic translation. The function of elongation factor Tu in the case of eukaryotic translation is carried out by elongation factor 1 alpha. The function of elongation factor Ts in the case of eukaryotes is carried out by elongation factor 1 beta, and the function of elongation factor G is carried out by elongation factor 2. These are the few differences between prokaryotic and eukaryotic translation. In the remaining things, the mechanism of translation is same in both prokaryotes and eukaryotes. One of the most important similarity among the prokaryotic and eukaryotic translations is the formation of polysomes or polyribosomes. Let us discuss what is this polysome or polyribosome. The other name is polyribosome. What is this polysome or polyribosome? In translation, an mRNA molecule involves which is of 5' to 3' direction. During the process of translation, what happens? The ribosomal molecules moves on the mRNA molecule in 5' to 3' direction, reading the triplet codons of mRNA molecule in order to synthesize a polypeptide chain of a specific sequence of amino acids. Before the completion of translation by this ribosomal complex, another ribosomal complex will come and attach to this mRNA molecule in order to perform the translation process to produce the same polypeptide chain again by reading the single mRNA molecule. Likewise, several ribosomes will come and attach to this single mRNA molecule in order to synthesize the several polypeptide chains, a single type of several polypeptide chains by using this single mRNA molecule. Thereby, a complex will be formed having mRNA molecule and a chain of ribosomes and polypeptide chains of different lengths. This mRNA molecule and chain of ribosomes showing a polypeptide chains of different lengths is said to be as a polysome or polyribosome. Here the polypeptide chain at 5' end is shorter, whereas the polypeptide chain at 3' end is longer. Keep remember this. Now let us discuss the last concept on the central dogma of molecular biology, that is post-translational modifications. We have discussed the post-transcriptional modifications after the completion of a transcription process. Likewise, after the completion of translation process, whatever the polypeptide chain being synthesized, it must undergo some post-translation modifications in order to become an active enzyme. Let us discuss those post-translational modifications. During translation, what happens? An mRNA molecule of a specific sequence of triplet codons undergoes a translation, right? After translation, a polypeptide chain will be synthesized of a specific sequence of amino acids according to the sequence of triplet codons present on the mRNA molecule. Okay, this polypeptide chain is actually biologically inactive. In order to become an active protein or an enzyme, it must undergo post-translational modifications, during which the polypeptide chain undergoes some conformational changes in order to become an active enzyme. Let us see those conformational changes or modifications that are being occurs at post-translational modifications. The first event in post-translational modifications is removal of, what is this? Removal of amino and carboxy terminal amino acids of a polypeptide chain. This is the amino end of a polypeptide chain. This is the carboxylic end of a polypeptide chain. During these post-translational modifications, the initiating amino acid at a amino end, that is formyl methionine or methionine in the case of eukaryotes, that's been removed, and the carboxy terminal amino acids also gets removed in these post-translational modifications. Because of this reason, we should not consider the initiating amino acid as the first amino acid in a polypeptide chain. Numbering starts from the second amino acids. Keep remember this. And the second event of post-translational modifications is modifications of individual amino acids. Say suppose for example, some amino acids of this polypeptide chain may undergo some changes. Suppose for example, the amino acids, say this is serine and this is threonine. These amino acids undergo phosphorylation, nothing but a phosphate group gets added to this serine and threonine. This is what a modification of individual amino acids, example phosphorylation of amino acids. The third event in post-translational modifications is glycosylation. Glycosylation, nothing but addition of a carbohydrate side chain to these polypeptide chains. These type of proteins we can observe on the cell membrane. The cell membrane consists glycosylated proteins. So this is a polypeptide chain. After glycolysis, it consists a carbohydrate side chains like this. This is what we call glycosylation. You know very well the process of glycosylation takes place in Golgi complex. And if this polypeptide chain, if it wants to become an active enzyme, it must undergo a post-translational modification, that is addition of prosthetic groups. What is a prosthetic group? It is a non-protein part of enzyme. Enzyme consists of a hollow enzyme consists of a protein part and a non-protein part. That non-protein part is said to be as a prosthetic group. That prosthetic group is being added to this a protein part of enzyme during post-translational modifications. So this is addition of this non-protein part. This polypeptide chain will become an active enzyme or hollow enzyme. And lastly, this linear chain of a polypeptide undergoes some conformational changes in order to get a three-dimensional structure. A polypeptide chain, whenever it gets a three-dimensional structure, it will have a specific active sites for a specific substrates. Say suppose for example, this is the linear chain of a polypeptide chain which undergoes a 3D structure like this. What happens with this? A specific sequence of amino acids will form an active site. This active site is a specific for a particular substrate. The conclusion of this post-translational modifications is to make a linear polypeptide chain, inactive polypeptide chain, to an active enzyme or active protein molecule to make it involved in a specific biochemical reactions. With this, the central dogma of molecular biology is over. In the next video, we are going to discuss the gene regulation with the example of lacoperon. Thanks for watching this video. Please like and share the videos and subscribe my channel Vidya Amrutam Ashnite.